to see we have some people for our inaugural seminar series. So I'm a group leader at Harvard Center for Advanced Imaging. I won't say anything else about myself because I really am delighted to introduce our main speaker today, so Tom Brokaw. So Tom has a very illustrious career that spreads over many, many different topics. He did his PhD in Stanford with Rob Lemon. And that's where he was really one of the first people to study these DNA recombination and replication intermediates of T4 bacteriophage. He did his postdoc with Norman Davidson in Caltech, and he actually invented, along with some other folks, the use of avidin biotin complexes to label DNA and RNA and proteins. Now, this was really the first time we were able to visualize specific genomic loci. In '75, he joined Cold Spring Harbor as the head of the EM Electron Microscopy lab, along with his wife, Louise Shao. And they continued to do EM work on the structure of DNA and gene organization and bacteriophage. He also later worked on R-loop and adenovirus organization. And that's when, along with his wife, they essentially discovered split genes and RNA splicing. Later, in '79, he became a tenured professor at Cold Spring Harbor, and then moved to University of Rochester and became a full professor. After this, he found his resting place at University of Alabama as a full professor, and really continuing and now working more specifically on HPV. He also is very important in terms of diagnostics. So Tom invented the test that is used for HPV genotyping that's not now sold by Roche. And really, I think for the last 30 years, HPV has been central to his work, studying how this also leads to things like neoplastic cancers and a variety of other issues. Tom also is a sort of a pioneer in trying to bring communities together. He leads the International Papillomavirus Society, which is going to have its 34th conference, maybe not this year, but as soon as this coronavirus is over. So speaking of that, Tom's talk today now is all going to be about DNA structure, organization, and essentially a broad history lesson on the fundamentals of sort of imaging of cells. So Tom, please take the floor, and we're very excited to hear what you have to say.
Thank you, Andrew, and thank you, Quasi. It's really a pleasure and an honor and an exciting opportunity to launch this seminar series. Just one small correction. I was the founding president of the society and was so for about 13 years, but I've stepped down. I'm on the board of directors at this point for the International Papillomavirus Society. So good afternoon, everyone, and thank you so much for this opportunity to speak, and I hope to interact and talk personally with many of you either at the end of the talk today or out into the future. What I really want to talk about is the powerful use of transmission electron microscopy to look at nucleic acids, DNA and RNA and chromosomes to try to understand their structural organization, their sequence features, and how they function in terms of transcription, replication, recombination. And I'm going to take 4 or 5 short stories that both I and Louise have worked on for our over 55-year career, almost all of it involving either EM or fluorescence light microscopy imaging. So to start, and I hope my commands work. Okay, this takes us back to our first few years at Cold Spring Harbor Lab, where we had a Zeiss scope and a Philips microscope. I want to introduce Louise, my wife, to you right now. And the lab chief in many ways was Emden Meyerhoff. So that's where much of what I'll be talking about today started. I want to take us back to, hard to believe, almost 50 years ago. What were our capabilities and what couldn't we do? Recombinant DNA technology was just barely getting off the ground with cloning. The first restriction enzymes were being discovered. DNA sequencing was limited to a few nucleotides at a time, incredibly short and difficult. No RNA sequencing. Gel electrophoresis was a relatively new technique. There were very few biochemical reagents that you could purchase, and essentially no kits because we hadn't invented them yet. PCR and RT-PCR were 14 years in the future. And all the imaging that we did up until about year 2000 required photographic film. That meant changing the film out every 12 pictures in an electron microscope. No digital imaging had come along yet. So how do we study genomes without any of those tools that we take for granted? Well, here's the problem. Genomes, as we know them now, span 6 orders of magnitude in length, from the smallest viruses that have a genome of several thousand base pairs, papilloma almost 8,000, adenovirus 35,000, bacteriophage mu 38,000, phage T4, which you're going to see quite a bit about, 168,000. Here's T4 attached to the outside of E. coli in a micrograph I did. It's 4.6 million base pairs, with the virus being 170,000 coming in and taking over this, this bacterium. And then we jump all the way up to us. Just our haploid genome is 3.2 billion. So 6 orders of magnitude. And it was essentially impossible to purify homogeneous genomes once you got past the point of the bacteriophage. And so everything to really understand the totality of a genome was limited to studying viruses. And this was the golden age of virology. Why do we study viruses? They're small, they're uniform length, and so they offer homogeneity. And so if you're doing biophysical characterization, if you have a preparation, it's the same throughout. It's not fragmented DNA. As we're going to see, and as many of you already know, viruses have very, very different molecular forms. They can be circular, they can be linear, they can be in several shorter linear fragments, but they all work. And they have very many different strategies for replicating their genomes. So there was a wealth of opportunity for learning about how replication and gene expression and regulation occur. So the phages really came into their own with Lurie and Delbrooks in the 1940s. And much of that learning and training was done at Cold Spring Harbor Laboratory. By the 1970s, it became possible to culture mammalian cells and therefore to study mammalian viruses. So this is coming up about the same time that our careers began. Most importantly, viruses depend on the host cell machinery for essentially everything they do. So they become both a probe and a tool to understand the next higher level, whether it's bacterial events or eukaryotic cell events. And of course, viruses, as we more than know today, cause diseases, some of them really terrible. And we need to understand them to control them, to diagnose them. But they can also be harnessed as vectors to treat diseases. So there was a lot of reason to understand these things. So I want to go in a few sort of background slides to talk about basic biophysical chemistry of nucleic acids. DNA duplexes, RNA duplexes, and the RNA-DNA hybrid duplexes can all be denatured or melted into single-stranded polynucleotides, for example, by heating. They can also be paired back together. If you mix two different genomes that are related but not identical, they'll partly pair and partly not pair. So this gives us an opportunity to look at sequence similarity and identity. And when you do that, you can make structures called heteroduplexes, mixed between two kinds of nucleic acid strands. And you can identify the location, the length, and many of the other properties of insertions, deletions, sequence duplications, inversions. And that's what we're going to talk about at the beginning. And so they have taught us a huge amount about genome organization, expression, regulation, mutations, and also precise phylogenetic relations among different species. And so I want to show some examples, and this is all designed to set the table for the talks that will follow using much more contemporary technologies. But in many of the principles were laid down 40 and 50 years ago. So a couple of technical factors, but I think they're very important whether you're doing electron microscopy or PCR or almost any other kind of in situ hybridization studies and so forth. Melting temperature is the temperature under certain conditions at which duplex nucleic acids can melt. And there's 6 variables that determine what the melting temperature is going to be. AT versus GC base composition and distribution in the strands. Are the duplexes RNA or DNA or a mix of RNA and DNA? What is the salt concentration? Have you put in what are called chaotropic agents that disrupt water structure? And these are typically formamide and urea that are agents that will break up water structure. And in doing so, they change the solvation and solubility issues of single-stranded RNA and DNA. Another factor in calculating the melting temperature is how long is the duplex that you're expecting, and is it near the end of the strand or is it in the middle? And also, is there a mismatch between the base pairs? For example, if you have 2 related genomes and you hybridize them to each other, but they aren't quite fully base paired, how do you calculate the degree of mismatch? Well, there's very precise equations that were worked out at Caltech primarily in the 1960s that, including by my mentor, Norman Davidson. Now, in reverse, if you can calculate the melting temperature of a duplex on an EM grid, you can actually reverse calculate the degree of homology or mismatch. And that became essential when I designed the first use of PCR to detect infectious diseases, because we didn't know exactly what disease we were going to be looking for. But by calculating the TM, I could find PCR primer locations that would work no matter what. And then a final term that you'll often hear in the literature is stringency. That means how much have you challenged a duplex to stay paired, or how close have you taken it to the point where it will dissociate? And high stringency are duplexes that will stay paired even 10 degrees below the melting temperature. 35 degrees, if they start denaturing, they're only about 60 degrees. So as you cool things, are they remaining, paired or not? Okay. Any manipulation of RNA and DNA involves 4 things that you have to calculate. And this really is the heart and soul of being successful at imaging nucleic acids. melting temperature is a thermodynamic parameter. And the other important factor is kinetics. So these are two of the fundamental principles of physics and chemistry, kinetics and thermodynamics. Kinetic factors primarily relate to concentrations, in this case of complementary nucleic acid sequences. There's other factors that do matter. Steric hindrance, if you have a very, very long nucleic acid, it can collapse into a ball, and the inner part of the ball is really not seen, and so it's at a lower concentration. And that relates to what's called excluded volume. And then finally, you have to maintain the integrity of the sequences, the strands of DNA and RNA without breakage. So you can control all four of these things, you can succeed at experiment. And then finally, one more thing. Double-stranded RNA and DNA can be spread on a water surface. I wrote a little bit into the abstract by the so-called Kleinschmidt method, where you can actually embed the nucleic acids in a protein film, float it across a water surface, and pick it up onto an EM grid. But single-strand polynucleotide sections will collapse, self-base pairing in water. But if you add these chaotropic agents like formamide or urea, that will actually help the base solvation, and single-stranded nucleic acids can be stretched out to their normal length as though they were in a duplex. And then finally, if you go from 40% to 70% formamide, RNA DNA duplexes are more stable than the corresponding DNA duplexes. So you can actually slip a transcript into the genome from which it was transcribed initially and see where it is. And that's the setup for what I'll be talking about. So for applications, I want to indicate several distinct types of DNA recombination, a little bit about probe technologies for imaging, some heteroduplex mapping to illustrate how we were able to use it for genome organization, and then finally R-loop mapping that let us develop the transcription map of the adenovirus, and in the course of that work made the utterly remarkable discovery that adeno, and it turns out you'll see essentially all other eukaryotic genomes, don't have contiguous genes as you would have in bacteria or phage, but in fact split genes. And the only way you convert that genetic information into a messenger RNA is splice the RNA. So it's a long way to go, and I'm going to try to do it as quickly as I can. Okay, my EM studies in Bob Lehman's lab at Stanford focused on phage T4 recombination and replication, in which case doing it using electron microscopy, I was able to discover that there are recombination intermediates that have branches as two genomes are coming together. I was able to figure out what enzymes were involved by making mutants in the T4 and asking could they still recombine or not. And in the course of this, I realized that a process called branch migration was absolutely essential to establishing heteroduplex overlaps between the two recombining DNAs. And to do that, you need to spin the DNA around its long axis. So dynamics became a problem. And you're going to see this. So here is an example of a branched molecule. You can see the DNA, one DNA here, and there's a branch point here, and there's a bridge, and then there's another DNA, and there's another bridge, and another branch point. So this is not just a simple recombination. These two or three genomes are interacting over and over and invading each other. And I can tell you they lead to just incredibly big, complicated structures of branches on top of branches. But we had no idea this was going on until we actually could look at it. And when you use the Formamid method and extend the single strand regions, what you see is this is a single strand end of one genome. That's duplex here. At this point, it becomes single strand, and it's invading this duplex and probably laying down a hybrid duplex here between the two genomes, but displacing the corresponding sequence from where it was invading. And I saw this over and over with this invasion, laying down one duplex that's heteroduplex hybrid, and displacing the corresponding chain of the target genome. Here's another example, another one, and so forth. So this turns out to be absolutely normal in T4 recombination, to invade, to displace through branch migration, kick out the area that you were invading. It may be assimilated elsewhere into another molecule, or it may be just removed by exonucleases or endonucleases. And here's a really pretty example. Here's a branch point here, and you notice the single strand. Here's a branch point with another single-stranded invasion. The two sides of this loop are exactly the same length. So genetically, the two genomes are staying in synapse in harmony in terms of genetic position. But we're actually seeing this swap out of one stretch of one genome for the other genome. And the same thing is going on over here. Again, a single strand invasion, a single strand invasion. Both sides of this loop are the same length. So you know it's really playing by the genetic base pairing rules. Very briefly, to describe axial rotation, because DNA is double helical and each strand is twisted around the other strand, you can't just untwist them. Just imagine a ball of twine or a garden hose, something that is wrapped around something. You have to spin it in order to unwind it. And I'll leave this talk will be archived, so you'll be able to review things or go back to literature that I wrote. But fundamentally, both DNAs are spinning, and they actually spin one strand off on the other. And it can be done with single strand invasion as here, or it can even be done double strand. And it takes no energy, because for every base pair you break as you pull off one strand, you're making the corresponding base pair with the new strand. And that works either single stranded or double stranded. And so the amazing thing is recombination at this stage does not take energy. So to summarize everything, I was able to work out the breakage and rejoining pathway, where you start with two double strands that made a thick one and a thin one. You have a nick, a single strand break. The single strand break is elongated to make gaps. Those gaps find each other through homologous base pairing. Usually in T4, it involves a single-stranded DNA binding protein. So you get the synapse, branch migration, then branch elimination, as I said, by either breaking off, cutting off, or converting it to a second molecule. And then you have to repair the gaps and seal the nicks. And the enzymes involved are endonucleases, exonucleases, single-strand DNA binding protein, DNA polymerases, DNA ligases, probably DNA primase, and topoisomerases. And I was able to work this out because using all the different available T4 mutants, I could stop the process at any point by having a mutation in that corresponding enzyme. So I knew how far it could go without certain enzymes. So all of that was published. I'll just say briefly that we also realized that that circular genomes could also interact, and either integrate into each other or excise out of each other. So almost all kinds of joining of molecules can be accounted for by the basic process I described, but always including branch migration. Well, in the course of this, I realized one more thing, because we were looking also at DNA replication structures. And as I already showed, the ends of all of these invading molecules were single-stranded. So I was asking, where did they come from? People thought it was exonucleases eroding back the end. But I realized it actually was being generated in DNA replication. You've heard probably of the so-called end replication problem, where you can't copy the very 5' end because you don't have a primer to copy that last little bit if the sequence polarity is going this way. So it turns out the ends of T4 replication are always left single-stranded. And that becomes the invading single strand for DNA recombination. So work done by my friend and colleague Gisela Mosig at Vanderbilt showed that replication forks are actually generated by recombination branches. So we kind of put our two themes together. So all of that's written up. And of course, this became the question of how do you replicate the ends of big linear chromosomes that led to Carol Dreider and Liz Blackburn discovering telomerase as an enzyme system that repairs the ends of eukaryotic genomes. But this was the first visual demonstration of what the problem was. So my postdoc work at Norman Davidson's lab at Caltech, which by the way is where I met Louise, briefly invented, it turns out simultaneously with two other labs, avid and biotin coupling techniques. And I'll show you two examples of how this was applied. References will always be provided. So my good friend Dave Hershey was a chemist, and he made polymethacrylate beads. Today we would call them nanoparticles. This is 1972. So nanoparticles have been around a long time. Dave made them. And this is what they look like for scale. They're about the size of a papillomavirus genome, virion. But what another colleague, Jerry Manning, did is he was interested in mapping the ribosomal RNA of Drosophila. So here's a typical cytogenetic phase contrast image of a Drosophila chromosome. This happens to be the X chromosome of polytene salivary gland Drosophila chromosomes. And at the base of that is the nucleolus. Now, of course, the nucleolus is where ribosomal RNA is made. It's a sort of a special body adjacent to or connected to the chromosome, but kind of spills out because it's so busy and active. So what Jerry did is he took Dave's beads, he took my avid and biotin coupling technology, we made avid and linked beads. Jerry took this picture, Maria and Jerry took the picture. Then they took the same microscope slide, denatured it, hybridized ribosomal RNA to the nucleolus, except now the ribosomal RNA was biotin tagged. And then they added the beads. And then they converted that same slide into scanning EM. And what you see here, this is the region of the chromosome right here in this box in scanning EM. Here's the nucleolus right there, and it's loaded with beads. So it was for the first time possible to actually see messenger RNAs hybridized to the array of ribosomal RNA genes. Well, that was mass production. What I did, and again, working now with Louise as well, we took a transducing phage that carried This is the wild type arm of the transducing phage in a heteroduplex. The other arm was transducing a tyrosine tRNA gene encoded by E. coli. So this side is a piece of E. coli genome. I hybridized tyrosine tRNA, which was biotin labeled, to the DNA, and then I added ferritin, iron-rich ferritin molecules, which I had coupled to avidin. And so now you could see the exact position of a gene that coded for something only 74 nucleotides long. So we had absolute precision, it was highly reproducible. And so for the first time, we could visualize and map the exact position of a single tiny gene. But there were many other applications. It turns out adenovirus DNA has a protein bound to the 5' end of the genome, which plays a crucial role in replication, in fact, serving as a primase for the genome replication. What we didn't know is, was there a protein on both ends and at both ends at the same time? So again, I did biotin labeling of the protein, whatever protein might be there. And then I built a chain of avidin and ferritin, linking them through biotin bridges. I mixed it with the tagged adenogenome, and now you see the bridge of ferritin molecules connecting the two ends. The only way that could have happened is if both ends had a protein at the same time. So this kind of made the point. I'm going to change gears completely now and switch to heteroduplex mapping. I can simply say Louise is the world's expert on this, but I did some of it myself. So I'm talking about both of our work now. Imagine a genome that contains an inverted sequence repeat. So the sequence reads this way at C, and it reads this way at D, and they are not the same, they're opposite, but they can pair with each other, actually this way. So if you denature this molecule and allow it to snap back, self-pair, it will make what's called a stem loop, indicating when you see this structure held together at the base, these are inverted repeats. And one of the properties of some inverted repeats is they're actually meant to be that way, and they can recombine with each other because they have homology. They're the same sequence in opposite order. And if they recombine with each other, that whole segment in between will flip over, and it can flip back through recombination at inverted repeats. Now, if you take a molecule that hadn't flipped over and one that had flipped over, you denature them, hybridize them to each other, what you'll get is a loop, because now CD on the upper strand is opposite CD on the lower strand, and they of course are not complements, they're identical. So they won't pair, you'll see the loop and you know exactly where the invertible region was. So how does this work out? Well, there was a project first project in our Cold Spring Harbor lab together, where Louise and I and Dave Zipser's lab had two postdocs, Common and Comp. They were the mu experts, but working together, Louise was seeing structures like this in the electron microscope and realized that there was a segment of the mu genome capable of flipping to two orientations. And through some beautiful genetic analysis that Dietmar and Regina did, the amazing story is that in one orientation, when you're transcribing into this invertible region, you do the tail fiber gene, and that's the tail fiber that enables mu to infect E. coli strain C. But when you flip it, you get the opposite strand being transcribed, And that's another copy of the tail fiber gene, but it has specificity for E. coli K. And these are the pictures that, without going through the subtlety of them, really prove that the two genes were in a cassette that could invert through these inverted repeats and change the host specificity for the genome. Was one of the earliest examples, clear examples of genetic switching through a recombination event. In the course of that, Louise expanded her exploration and realized that there was also an invertible region in phage P1, which is quite a different phage. But she was able to hybridize one strand of phage mu and one strand of P1, and it turns out they pair with each other. That's that circular duplex right here in the middle of single-stranded regions. Here's the P1 strand. Here's phage mu with the so-called invertible loop itself already opened up. Tracing, it looks incredibly complicated, but if as Louise did, knew exactly how to interpret these things, it could make sense. And it turns out that mu and P1 share the exact same invertible interior genetic region for host range variation between E. coli K and C. Well, it turns out, interestingly enough, the P1 version of this, what's inside that invertible region is the Cre enzyme, a recombinase that acts on the two inverted repeats to recombine them. And the recombination site is called LOXP, and many of you are aware of the Cre-LOXP system for engineering living creatures to turn genes on or off in, say, early development of a mouse or Drosophila. So it turns out this is the beginning of that. The Cre-LOX system is in fact encoded in this loop area, and that enzyme acts on the ends of the inverted repeat. So this all goes back to 1976. I'm not going to make much of a point here, but it turns out a structure that I came across over and over were strange double lollipop systems. That's basically a cruciform, which is a recombination branch point in many ways, but we made them through heteroduplexes. And it turns out if these two loops form the stable stem, very stable stem, and then the outer flanks pair, the loops can't wind around each other in a double helical way, but what they'll do is they'll try to hybridize and they'll form what I called an underwound loop. They're trying to pair, they can't, but we came to realize that these underwound loops were absolutely diagnostic of inverted repeats. And we were able to go on and find many, many such examples. One of those examples turns out to be insertion sequence 5, a bacterial insertion sequence. This was the heyday of jumping genes. Our immediate next door neighbor at Cold Spring Harbor was Barbara McClintock, who of course won the Nobel Prize for jumping genes. But the bottom line is we could now start visualizing these. These are various insertion sequence 5 and 2 that were being identified genetically, but we were the first to, I think, really image them. And it turns out IS5 makes that underwound loop. There's the phage mu invertible loop. It also is a transposon system. And other loops of these underwound loops. So we were able to quickly see other underwound, other inverted repeat sequences based on these underwound loops. They really jump out at you. And so this led to a variety of studies of transposons and insertion sequences as well. So again, those are all forms of recombination. So now I want to go on to a new set of questions. what were the big questions about chromosomes in 1972? Why are their genomes, why are our genomes so large? Why do we have 3.2 billion base pairs? That's a whole lot of genetic information, potentially a whole lot of genes. How are the genes organized in those chromosomes? Do we have, in a sense, a million genes? We didn't know for sure. Do eukaryotic cells, plants or Drosophila or us, do we have operons like bacteria do, with coordinated expression of a set of nearby genes? What happens to eukaryotic messenger RNA after it's made? And what I'll point out is that many of these answers actually came from direct EM visualization of viral genomes, which were then applied to eukaryotic genomes. So let's go back to 1949. Beadle and Tatum, based on genetics, came up with a concept of one gene makes one protein. Got the Nobel Prize for it. And of course, over the next 20 years, with the discovery of DNA structure, RNA, the messenger RNA hypothesis, and then studies that showed that there was a direct correlation between the RNA that is made and the protein that is translated from it, we came up with the central dogma of molecular biology. One gene makes one RNA, makes one protein. The collinearity was so simple and so straightforward and so deceptive. So now I want to go back to how we address those questions. In 1976, one of my former professors at Stanford, Dave Hogness, and my also advisor, Norman Davidson at Caltech, independently came up with an observation that RNA DNA duplexes are more stable than the corresponding DNA duplex of the exact same sequence if the solution is in 70% formamide or higher. It's just a strange anomaly. But what it meant was you could insinuate an RNA transcript back into your gene of origin, displacing the homologous strand, and those things are called R loops. And this is what one looks like. I worked with Tom Maniotis on this one. He had cloned the beta globin gene and then made a beta globin mature RNA preparation. And I was able to use this to make our first R loop, which was, here's the vector of DNA in which the globin gene had been cloned. And then this side is an RNA DNA hybrid, and this is the displaced single strand. And so you could actually slip the RNA into this. Now, the irony of all of this is the gene here was not the natural gene. It was a cDNA copy of the messenger RNA. And that in a way was tragic, because if it had been the original gene, we would have discovered splicing 1 year earlier. But we applied this to the adenovirus genome, something that had never been attempted. This is 35,000 base pair long genome from the left end all the way to the right end. And we mixed it with a very good quality preparation of adenovirus RNA that our good friend Jim Lewis had made. And here you see the R loop for the hexon gene, major capsid gene, the fiber gene R loop, the poly A tail, and then another structural accessory protein called 100K, a third R loop. So we actually realized you could map the positions of individual RNAs. And we looked at thousands of these things. And remember, only 12 photographs per load of film in the microscope. Every time you have 12 shots, you empty the film out, re-evacuate the column with new film. So we did a lot of this, and we used early RNA preps and late RNA preps. And here's, there were 5 early regions of adeno. Here's early region 1A, early region 2, early region 3, early region 4, all in the same molecule. So we can measure their positions and their lengths. And early region 1A actually has 2 parts, 1A and 1B. Here's 1A, the same as here, but here's 1B. So you have all these regions, and what we were able to do then is mix late RNA and early RNA at the same time and see what are called strand switches. So I'll take you through this one. Here's a hexon R loop, double-stranded RNA DNA hybrid, and at this point it goes single-stranded. It's thinner and knobbier. But on the corresponding strand on the other side is a duplex up to this point, and then it goes thinner and knobbier. And that is a strand switch between the hexon RNA and the single-stranded binding protein gene, 72K. And each of these is showing a strand switch between early and late transcripts. So generally, one will be going one way, the other will be going on the other strand, and they meet in the middle, and they both stop at that termination site. So what we did is we took literally thousands of these, this is a small subset. We lined them up and asked, and this happens to be the hexon gene, and this is the fiber gene, and this is the 100K gene, and here's early region 1A and 1B and 2 and 3 and 4. And we could line all of these up. This is the 5 early regions and all the late regions, and figure out which strand was making what RNA, and work out the whole map. And this is what it looked like. The early genes going this way, late genes coming that way. All these late genes transcribe this direction from left to right. Some of the early genes coming from right to left. And very complicated. But we laid them all down. And I want to point out that Mike Matthews at Cold Spring Harbor had been studying very small RNAs called viral associated. They're here. And upstream of all of these transcripts. And that becomes very, very important. These maps were also correlated with DNA restriction cutting maps that Rich Roberts and others had developed, Phil Sharp's lab and so forth, when he was at Cold Spring Harbor. So we had a really good map. Now, one thing we observed when we were doing this, because we were so familiar with these single-stranded branches, we were seeing them at a lot of these R loops. Now, some of them made sense to us as poly A sites at the 3' end, but we were seeing branches at the 5' ends. There's an example there. Here's one here. And the question is, why are these single-stranded branches present? So it turns out our colleagues, Rich Roberts and his staff scientist, Rich Gelinas, were looking at the 5' ends of adeno late RNAs. They were capped, and they all were. This capping was a very new phenomenon from Aaron Shatkin's lab in particular around 1972. But what Roberts and Gelinas found is they seem to have a very similar, if not identical, sequence, no matter what late RNA they were attached to. And that suggested to them a hypothesis and an experiment. Now, here's the hypothesis. A year earlier, Dixon and Robertson at Rockefeller had shown that influenza virus, when it infects, breaks down host RNA into fragments, and then it hijacks random fragments to prime the synthesis of influenza RNA transcripts. And that was published right about this time, and Hugh and Rich discussed this issue. So Roberts became convinced that the VA RNA that I had just shown you served as the primer for all the RNAs downstream of that point, namely every one of these messages that we had been mapping individually, like the hexon RNA, 100K RNA, the fiber RNA, that they were primed by the VA fragment in the following way. The primer lays down on the genome and extends to make that transcript. Another one pairs at a direct repeat somewhere further down the genome and is extended, and so forth. And that is how all the RNAs are initiated in adeno. Well, here's the experiment that in a sense, they more or less suggested that if the 5' tails included the 5' capped sequence, that might be VA RNA. So we made our loops. We always would be able to see the 3' poly-A site. But what we knew is you had to use separated strands of the restriction fragments that we're going to try to hybridize. Hybridize a restriction fragment spanning the VA gene right here to the complement, and then it would be the primer of that message. So this required really good RNA from Lewis, our knowledge of the map coordinates of all the late RNAs and that we had seen, and then everything I talked about in the second or third slide, which was how do you calculate TM? We anticipated this stretch could be as short as 20 nucleotide base pairing, maybe a little longer. We didn't know. But because I really had become an expert in melting temperature, I was able to anticipate how we could assure that these two RNA, this RNA, this primer could hybridize to each other. So we worked it out and it's published. And guess what? The experiment worked. We absolutely got tagging of the 5' end of the messenger RNA. Here's hexon. However, it wasn't VA RNA. It wasn't even close. And it was actually 3 points of contact with the 5' end with the restriction fragment, not 1 point of contact. And none of the points corresponded to, even close to the VA gene. So VA priming was not the answer. Adeno was doing something different. And we looked at more and more of these and found we could decorate the 5' ends with various restriction fragments, but they always form this same multi-loop structure. You see a small loop, a bigger loop, a small loop, a bigger loop. Over here, a small loop, a bigger loop. And we did this over and over, hundreds and hundreds of examples. And in the course of this, we found other structures that were nothing like them, but they made loops. And one of them was the 4A2 RNA right here with this tiny little blip. Here was another one from an early region, making a pairing, and then a loop, and then some more pairing, and so forth. And so we were seeing a phenomenon that just made absolutely no sense. Well, it turns out it was split genes and RNA splicing. And of course, we're all familiar with it now. A long transcript is made, it spans a very long gene, sometimes a shorter gene, but it's processed at the RNA level to keep certain fragments and delete other fragments and recombine them. So again, recombination, something we'd worked on for our whole life, except now it's RNA recombination. And it turned out there was a 3-part leader on the end of all these late adenovirus messages. And this is sort of a diagram of them. They came to be known as exons and introns. And here's one example on the cover of Cell. When we published this, there's a decorated end on hexon and a decorated end on the fiber gene. And so that was the really, really big breakthrough. And we published in collaboration with 3 other groups at Cold Spring Harbor that were getting biochemical information supporting the same thing, presented it at the 1977 symposium appropriately enough on chromatin. Totally transformed the field. I had one of the participants in that meeting on chromatin was Francis Crick. And when he heard my talk, I guess he was both astonished and perplexed. And so we had lunch together. And I really had the privilege of explaining to Francis Crick what RNA splicing and split genes were. And that was actually the pick. So Louise and I continued to study this very, very exhaustively, and we were able to find all kinds of splicing intermediates. Here's an example of a fiber, not only with the 3-part leader, but actually 8 parts to it, with stuff that were exons that were ultimately going to be taken out as introns. And all of these different splicing intermediates were found just for the fiber message, as one example. Sometimes it has 6 leaders, 1, 2, 3, X, Y, Z. But we worked out a whole lot of that. And after 5 really hard years, developed the adenovirus transcription map. And what was really, and meanwhile, Jeff Engler in our lab started sequencing adeno, and the area we wanted to look at was from coordinate 10 to 30, because they're encoding a new set of transcripts that nobody knew about. What were they? And so this is Jeff's sequence, and it shows how we could correlate the EM and sequencing now that it finally, this took 2 years to do. Finally became available. And here's the various proteins. There's spliced 4A2, the 3-part leader, and a 4th ancillary intermediate leader for the late RNAs is here. Here's the VA-RNA genes coding region, and then various open reading frames. And this is the terminal protein gene where I had labeled that protein with ferritin in the earlier picture. And here's the adenovirus DNA polymerase. And one of the early region 2s is the single strand binding protein. So we were combining our interest in recombination, transcription, and replication. And this one sequence kind of summarizes it.
Tom, just a quick prompt that we're getting out of time. So
yeah, okay. I'll wrap it up real quick then. I'm actually right about at the end. We took all the sequence information and kept looking at spliced patterns, and ultimately figured out a huge variety of things that splicing achieves, bypassing stop codons, frame shifting, making alternative variants of a single protein, building up operons of multiple proteins that work together, and so forth. And so this is a diagram that I stole from Michelangelo, a 3-part leader joined to the chosen messenger. So I'll wrap it up by saying Louise then collaborated with a zillion other people and mapped not only viral genes, but all of these host genomes. All the references are here: tubulins, collagen gene, prolohydroxylases, genes associated with genetic diseases, laminins. And just one example done by EM in collaboration with the Tryggvason lab in Finland. All of these exons and their intron spacing, all done by EM, but did that with all those other genes as well. So the radically new dogma that replaced Beadle and Tatum is one gene, many enzymes, or many genetic segments combined into one gene. So E pluribus unum, X pluribus una. Alternative promoters for transcription, alternative polyase sites. I think this is the biggest part of the discovery. Alternative splicing that generates a great variety of products from a single genome. And this then raised all kinds of questions on regulation, on changes in gene expression during development and differentiation. And of course, it's now known many genetic diseases, starting with the Alport disease that Louise worked on. And to wrap it up, we did the same thing, moved on to human papillomavirus in 1980. This is its transcription map, really complicated, multiple promoters, common and variable polya sites, internal splicing all over the place. And we spent 40 years studying this. So I'll wrap it up there and say thank you for your attention. And I'm certainly happy to take any questions you might have, or you can communicate with me later.